Hi, everybody. My name is Angelica Vazquez Ortega. I am a professor at Bowling Green State University. My area of specialization is geochemistry. And I'm going to be presenting my research to you guys. And the title for this presentation is Assessing the Impact of Agricultural Best Management Practices on Crop Yield, Soil Health, and Water Quality across spatial and temporal scales. This is pretty much the research that I have done at BGSU during my tenure track. So you will learn a lot of different research areas that I have been doing, but all of them has a common theme. So let's start. Okay, I'm going to start providing you some background information to understand the relevancy of my research and what has been the motivators behind the research that I have been conducted at BGSU for the last 5.5 years. So first of all, I want to start with a very relevant and important problem that we have in the area, which is that we have been experiencing, or our Lake Erie has been experiencing harmful algal blooms for the last 1 or 2 decades. And these algae bloom, or HAPs in short, they pose very detrimental effects on the health of the lake, including the fish in the area. Also, it's detrimental to the human health. And many communities around the area are being affected. The economy of the area is being affected. For instance, farmers that are fishing in the area, their business has been affected, and also houses in the area have lost their value in the area. So in most instances, the formation of these algae blooms has been correlated with agricultural runoff pollution from the area. And it can be in the form of synthetic fertilizers or manure fertilizers. So this map that I'm showing here corresponds to the watershed of the Maumee River. So it's the basin of the Maumee River, and I'm trying to find the laser here. So it is part of Indiana and also a large part of Ohio. And here will be the Lake Erie in this position. So this specific map, it's presenting the land use or land cover of the Maumee basin. So you will notice that in yellow, it's the land use regarding for agricultural area, agricultural productivity. In specific, the main products or the main crops or cash crops, as they are known, are corn, soybeans, and to a lower degree or extent wheat. So we can appreciate from this map that a very large amount of the area it's being occupied or it's being used by farming. So when you have a watershed that is so that farming is so prevalent, even though farmers are doing their best job on putting the minimum amount of fertilizers, it's inevitable that a portion of those fertilizers will be exported into the lake. So I am working, and I'm not the only one. There are many different universities, as well as let's say, for instance, soil and water conservation districts, the Ohio EPA, and other agencies that are working very hard with farmers to make sure that the export of nutrients is minimal. It's minimal. So one characteristic of the area is that there is a lot of tile drainage in the region. And this is very important to understand, because this means that typically we can think of the soil as the filter, okay? It's filtering nutrients, it's filtering water and potential pollutants within that water. Because soils have the capacity to sequester or to absorb very tightly different species, different components. For example, pesticides can be trapped and then degraded by bacteria. Nutrients can be sequestered, or they can be, for instance, nitrogen. During the nitrogen cycle, you can have nitrate going into nitrogen gas and being escaped into the atmosphere. However, when we have this type of tile drainage, which is necessary, if we look at this image here, we can observe that in this region, in the Northwest region, we also know that we are part of the former Great Black Swamp. And in order for us to use the fertile organic rich soils in the area, we need to drain the wetland. And this is the way that we do it here in Ohio, in which you have the water table very high due to the wetland conditions. But then when this type of a structure is put in place, a tile drainage, then the soil can be drained. And the root system in this instance, for example, corn, they can grow very robust, and then we can actually harvest a good yield. And the same example for soybean and wheat, the root system needs to be aerated. And when you have high concentration of water or water saturation, that's not possible. And then we hinder the growth of the crop. But the implications of having these type of systems is that now the function of the soil in sequestering or retaining or cycling the nutrients, it's being hindered. So now all of the nutrients are kind of like bypassing that soil and entering into the waterways. So the map that I have here, it's a very nice map showing the degree of tile drainage in the area, in specific in the United States for 2017. We can here observe in the legend that the more, if we move more towards the blue colors, that means that those areas are very heavily tiled, okay? And we can observe that that's the case here in this area, which is also known as the Corn Belt. So here we have Ohio, here we have the Lake Erie, and we can see that the northwest part of Ohio, which also corresponds to the Maumee Basin, has been heavily tiled. So when we have a combination of both, we have a very heavily tiled watershed, and pretty much 85% of the land use is farm. And then even though we have a small amount of nutrients, when you multiply that by the whole area, it's a significant amount of nutrients that are reaching our lake. And Lake Erie is a shallow and warm lake. And usually by the end of summer, let's say July, August, September, it's when we see this huge bloom in the algae, in HAPs, okay? So here in this map, in this other slide, I'm showing you guys different images of how the lake looked like when we have the bloom, okay? And here in these other figures, what I want to show you, it's a relatively strong correlation when we think about real data, experimental data in a natural system. So we have here in the x-axis, we have how much soluble reactive phosphorus in loading, so in terms of mass, how much it's being exported by the Maumee River. And that's being correlated with the mass of the cyanobacteria and also total phytoplankton or just cyanobacteria masses or biomass. So we can observe that as the export of this soluble reactive phosphorus increases, we are also seeing an increase in the biomass of the cyanobacteria. So there is a correlation between how much is being exported from the watershed and it's being received in the lake to the incidences of having algae bloom in the lake. So we can see how everything is, it's a beautiful puzzle that we can put it together regarding land use, nutrient export, and then the algae bloom that we are observing at the lake. So, but what can we do to minimize the export of these nutrients when we have such a vital and vibrant agricultural productivity in the area? Well, farmers are working very hard to put in practice different agricultural best management practices to control the nutrient loss. And these practices can be put in place in the field to minimize or to increase the cycle of the nutrients in the field to ensure that the plants are getting access to the nutrients, or at the case that those nutrients are being lost into, out of the field, then maybe they can be captured at the end of the field. So again, we can have in-field or edge of field practices, but these practices can also be separated or categorized by different types of management. For instance, we can have soil management that can manage directly the soil health or the soil texture. And those practices can be more like conservation tillage. Instead of tilling very aggressively, there can be minimal tillage or no tillage at all. Water management practices can also be used. For instance, we can use riparian buffers or wetlands to try to capture that water before it gets into the waterways and eventually into Lake Erie. And then finally, we have the nutrient management, which is a very important tool for farmers in the area, in which they can have a nutrient plan and have a better idea of when is the best time of the year, or when is the best time of putting that fertilizer, or at what concentration, what is the type of fertilizer that they should be putting in place in their land. So a combination of all of these BMPs, again, for short, best management practices, they can be used by farmers in the area to minimize the export of nutrients into the waterways. So now I'm going to be moving more towards one specific practice that I have been studying in my lab. And this one is the use of dredge sediments. And first, I want to explain why we are using dredge sediments and why they might be relevant as a potential BMP in the region. So first of all, we have in the south or the US portion of Lake Erie, we have 8 main ports. And those ports are very active in the area, and a lot of the economy is dependent on them. So we need to make sure that these ports are in good conditions for the ships to go in and out and keep this very vital economic activity in the area. So, but in order to do that and for the ships to navigate the area, these harbors needs to be dredged. And what I mean by that is the sediment at the bottom of the channel, they needs to be removed. And if we want to make a connection of first, why do we have to dredge then? Well, a lot, part of the geologic processes in the area, we know that we have soil erosion. So a lot of the top fertile soil that we have in our farms in the Maumee watershed, it's being eroded, okay, either by water means or soil, or I'm sorry, water or wind erosion. So that top portion of the farm soil, it's being eroded and ending at the lake in a specific at these very active channels. So when the channels are dredged before 2021, summer of 2021, the practice to get rid of those sediments is to dredge them, put them in a barge, move more towards the central area of Lake Erie, and then dump those sediments. And this is an example of it's a cartoon of a barge, but this is an example of how that process will happen, okay? So that barge, it's releasing those sediments, and whatever is in the sediments, either of course particulates or nutrients or pollutants, they are going to be redistributed in the water column. And that can exacerbate the algae bloom in the area if those sediments are very rich in nutrients. So with the government of Ohio, they came to the conclusion that they do not want this practice to occur anymore. So they have banned the open lake disposal of dredged sediments in Lake Erie, at least in the US portion of Lake Erie. And instead of using that, now, if you guys can follow my pointer here, you can observe this square island. It's a man-made island that has been exclusively designed to receive dredged sediments. So the Army Corps of Engineers manage this area with the Toledo Port Authority. And now, instead of discharging the dredge in the open lake area, in this specific rectangle, is the one that has been assigned. Now the dredge is being placed in this facility, in this confined facility. And the idea of placing this dredge in this area is to allow for the dredge to dewater. Pretty much 90% of the material that it's being removed from the bottom of the channels contains a huge amount of water, like 90% of water. So this island allows the water to be discharged and then the sediments being retained. And the idea is to allow for this maybe for 3 to 5 years for the dewatering process to occur. And then those sediments that are pretty much soil, like a topsoil, very fertile topsoil, to be used for different beneficial uses. And potential beneficial uses can be, let's use the sediments for construction, for instance. Let's use the sediments to create a wetland. But in the case of my investigation, I'm using the dredged sediments to amend a farm field. And it's like, think about it in a way of adding synthetic fertilizers or adding compost or manure or biosolids. But in this case, it's adding the sediments, okay? So you will ask yourself, is there a nutritional value in this sediment? And that's why I have this table here. So, so the table here, it's showing different parameters, comparing those parameters between the concentration in the dredged sediment versus the optimal values. And in this specific case, it's for the row crops that we have in the area, pretty much corn and soybean. So some of the parameters that I would like to emphasize here is, for instance, the cation exchange capacity. And we can observe that an optimal value might be around 21 in this specific unit, and the dredged sediment has 35. Another very important aspect is the bioavailable phosphorus that is within that material. So again, a typical farm soil, you want it to be between 15 to 40, more than 40 parts per million, it's not recommended. It's now we are moving on over fertilized soils or a term that has been coined in the area as legacy fields in which there is too much phosphorus in the soil. And you can see concentrations as high as 100 to 500 parts per million. So when a farmer wants to target an optimal value for phosphate, they want to target that range between 15 to 40. Less than that, it's too little, and that will be detrimental for the crop. Higher than that, there are not going to be any higher yields, but it will be very detrimental for the environment. And we can observe here that the dredge sediment can be between 40 to 80. So it's not extremely rich in nutrients, but it can provide an adequate amount of nutrients when the dredge sediment is added at a optimal concentration. Another parameter that I want to emphasize here is calcium. Here is the range, and see how rich in calcium are these sediments. Also, it can provide potassium, which I have heard from different farmers that potassium is very important, and farmers add potash. So these dredge sediments also can provide this essential element in this case. So again, my idea is, going back to this diagram here, it's to use the dredge sediments as a farm soil amendment. So it's a potential BMP, and if we go back here, it can be a potential BMP to manage the soil, because the dredge sediments are rich in organic matter, which will improve the soil structure. And it's also a way of managing the nutrients, because now by adding the sediments, the dredge sediments, which were before eroded fertile topsoil, now we are adding back the nutrients. So it's pretty much like closing the cycle. Like the farmers are losing this precious material, they're very rich and valuable topsoil. It's being lost into the lake. The lake now, those sediments are being collected, dewatered, and then we can bring them back to their farms. But the question that we have to ask is, we grow crops in dredge sediment, or what the concentrations, or the how much tons per acre, what is the rate that we have to put in order for this to be feasible? And that's what I'm going to be presenting you in the next slides. In addition, that I'm going to be also presenting other different research that I have done that are pretty much very they are very linked together. So I also want to show this figure here because it's very important, as this is the framework that I'm using for this presentation. So here I have different scales, and during my trajectory here at Bowling Green State University, I have been able to do research across all of these different scales. So some of the research that I have done, it's very, it's looking more at the solid mineral interface, then moving more towards the soil profile, and that has been done mainly on greenhouse experiments, in which I have different buckets that are a proxy for that soil profile or that mesocosm. And then I also have experiments at the field scale to test if what I am seeing in the greenhouse can also be projected into the field scale. And then finally, I'm also in collaboration with other researchers on doing experiments at the watershed scale, but also doing contributions within different soil profiles or field experiments within the watershed, big umbrella. So let's dig in on all of these different scales, and I will walk you through all the different research that I have been doing in my research team. Of course, I have been leading and advising my students. So at the end of the the main idea here is that everything has been done thanks to the students, the graduate students that have been working with me. Okay, so looking at that solid mineral scale, I want to show you or to discuss a little bit more about a research that Fatima has, that she did in my research. She already graduated and it's moving towards getting a PhD. So the title for this specific experiment is the role of dissolved organic matter of phosphorus sorption onto iron-enhanced activated aluminum media using flow-through column experiments. Okay, very long title, but I just want to summarize it very quickly here. So the idea with this experiment is to understand how dissolved organic matter might hinder the sequestration of phosphorus into this media that is being used in phosphorus filters at the edge of the field in different fields. And this is an area of research that has been done primarily by Dr. Shad Penn at the USDA. So, and I have been working in collaboration with him. And I'm gonna go back to this diagram in a second, but I wanna forward because I also wanna show you what are these phosphorus filters and why it's relevant to understand the role of organic matter in this case. So what I'm showing here are different ways in which we can build or construct these phosphorus filters. So they can be blind inlets, or they can be, this material can be placed at different waterways, or they can be put inside a type of septic tank. So there are different ways in which this material, a material that can absorb phosphorus, can be placed, and then the water, either tile drainage water or surface water, can move through this system and they will retain the phosphorus. So there are different materials that can be used, synthetic materials or metal scraps or whatever that can be used to fill these different structures. And then when you divert the water, that the water can move through these structures has such a high affinity with phosphate that it will be retained in the media, and the water that it's being, the effluent, now it is deprived of some portion of the phosphorus. So what Ume did is, and we are doing this in a laboratory scale. So we need to take these huge structures and put the media in smaller columns. So here are her columns filled with the media, and here it's a container, a solution containing both organic matter and as well as a specific phosphate concentration. So what she did is she's moving through the columns, the solution, and then she's collecting the solution in the fraction collector. And what she measured is at different concentrations. Here is this figure that she obtained from her research. Here we have, let's go with the bottom one. We have pore volume here in the x-axis, and then we have here the concentration of organic carbon at the effluent, okay? And what we observe is that at the beginning of the experiment, depending the concentration of the organic matter, so 5 ppm, 10 ppm, 20 ppm, we can observe them here. At the beginning of the irrigation into the media, a portion of the organic carbon was retained in the media. So what she wants to test is this organic matter, will the organic matter that is in the influence solution will be competing with the phosphate, and it will be hindering the efficiency of the media in retaining phosphate. And this is something very important because right now, when these engineers or scientists are designing this type of structures, they are not taking into consideration organic matter, the role of organic matter. And what Umed proved and tested is that here is the phosphate. If we think about, well, if we look here at the y-axis, it's how much phosphate was removed versus how much was put into the system. So what she observed is that here in the black squares, we can observe, over time, a huge amount of that phosphate was retained in the system. However, this first line has no organic matter at all. So at some point, the efficiency is decreasing. However, when she added the organic matter at different concentration, this efficiency even decreased even further. So she proved that organic matter is competing with the phosphate, decreasing the sorption of phosphate. And this has huge implications, again, for the design, because let's say the designers can say, okay, if you design this specific structure, it might have a lifetime of 10 years. Well, actually, it might be lower because something else is competing, and in this case, it's organic matter. So here are different examples of how that phosphate is being absorbed onto the surface of the media. And then here we have an example of, okay, I have that phosphate, but I also have an organic matter, and that organic matter is being represented by a carboxyl functional group here, and it can be competing. So bottom line for this specific research, this is a really nice BMP to use, this type of phosphorus filters, but we might have to take into consideration the soil organic matter competition when designing those different practices. Okay, so moving on to another research, another experiment that was conducted by Matthew Franks, again, a master's student of mine, again, being in that interface, solid mineral interface. What I want to show you here is an experiment that he conducted to study the role of iron and manganese oxyhydroxides on carbon and nutrient dynamics in agricultural soils. So again, to summarize this is these iron and manganese oxides are prevalent in the soils, and they are neoformed constantly in the soils. And they play a very important role sequestering carbon and nutrients. So what Matt wanted to do is, or what he did is, he conducted a chemical sequential extraction, which the parameters are here. And he was targeting different phases and using different solutions or reagents to try to do a reductive dissolution of the different minerals. And then what he did is he associated the content of phosphate and nitrate with those different mineral phases. So he had actual farm soils from different farms, and he exposed that soil to this extraction regime. And what he observed here, for example, we have the water extractable, the manganese oxide, amorphous iron, crystalline iron, and then the leftover, the residual. What he observed is that for phosphate, so here we have the mass fraction of phosphate, and here we have different soils as a function of depth. What he was able to learn is that these manganese oxides and iron oxides exert a very strong role on retaining those nutrients in the soil. We can see here the scale going from 0 to 1. So we can say that maybe 20% or more of the manganese oxides are retaining phosphate, okay? And we can see that trend for different fields that he studied. And I'm also only showing you guys here a couple because there's no time to show you all of them. So the bottom figure is for nitrate. And again, look at the scale. Here it's from 0 to not even 0.04. And we can observe how little influence this mineral has on the retention of nitrate. So if we are looking for BMPs that will make sure that these minerals are stable in the area, we can observe that these minerals are very, very important. Again, the manganese oxide and the iron oxides are extremely important in the retention of phosphate in the system. So we need to find BMPs that are going to be conductive for their stability. And one of, changing in water content, we can have anoxic conditions, and those anoxic conditions can induce the dissolution, the reductive dissolution of these minerals, and with that releasing the nutrients that are associated. So that's part of Matthew research. Let's keep going. Okay, again, within the same scale, solid mineral scale, I'm going to be presenting to you research that Samira and Katarina has been doing in my research. In this case, they are doing another chemical sequential extraction, but they are targeting humic substances. Let's read the title for the research. Characterization of supramolecular humic material obtained from lake dredge sediments and farm soil by humiomics and spectroscopy methods. So humiomics is just the name of this specific extraction that I'm showing here in this flow chart. And the idea is to think about organic matter not as a polymer, but as a supramolecular structure that has components that can be identified. And by going through this extraction, we can extract different portions, the unbound, the ester, and the ether. So Samira and Katarina are doing an excellent job going through this organic chemical extraction to try to separate the organic matter components within soils. And in this case, they are going to do it for both dredge sediments and for farm soils. And the idea is to develop this method and apply this to soils that has been exposed to different BMPs. For instance, a soil that has been heavily tilled and then changed to no tilling, and see how that organic matter changes. And not only how much organic matter is being accumulated, but what are the different types of organic compounds that are being accumulated. Here, this is an example of the different fractions that can be separated with this technique. And then again, what are the different compounds that each fraction it's able to remove. And so here, they also are trying to correlate with each fraction, how much nitrogen and how much phosphorus it's being retained. So we can have a really good idea of not only, again, how much carbon or what type of carbon, but also what is the role of these different type of organic compounds regarding the retention of nitrogen and phosphorus. And this is very important, again, because we are opening the black box now of the soil compartment, and now we are separating, like, okay, these fractions are very relevant and very important because they can contribute organic nitrogen or organic phosphorus that can be mineralized into phosphate and nitrate and be bioaccumulated by plants or used by plants. So finally, for the solid mineral interface, I want to show you research that Caroline Bart it's conducting with me and Dr. Kevin McClooney in biology. So what she is aiming is also to understand the phosphate cycling, but in this case, she is looking at the oxygen phosphate isotope signatures within her different dredge mixtures. So it's the biological cycling of phosphate in farm soils amended with dredge materials, obtaining insights from oxygen phosphate isotope signatures. So she's also doing a very complicated extraction and precipitation. Here we can see different examples of what she is extracting from this procedure. And then she developed a research in which she added synthetic fertilizer or she added heavy water, 2 different buckets containing different ratios of dredge sediments to farm soil, and she was growing corn. So what she wants to do is understand how the presence of dredge might help in the cycling of phosphate in these soils. Very interesting. Again, I'm just giving you very briefly information of all of this research. If you guys are very interested, you can contact me and I can put you in contact with my students, which are the experts in these specific topics. Okay, so now I want to move to another scale. Now it's the soil profile mesocosm scale. Okay, now I'm moving a little bit more larger in a scale. Now it's during different greenhouse experiments that we are seeing more regarding soil health and crop yields. So in this specific project named Assessing the Effects, and I'm going to show you within this topic, different experiments that have been conducted in my lab, assessing the effects of lake dredge sediments on soil health, looking for agricultural and environmental implications. This specific one, Russell Brigham, which also graduated, and Stephen is currently doing his research with me. He did a greenhouse experiment planting soybeans with different ratios of dredge and farm soil, 100% farm soil, 10 dredge, 90 soil, 20, 80, and 100% dredge. And what he, so he allowed the soybean to grow, and then he harvested that. He took soil samples, he took the grains, the soybean grains, and he analyzed a vast amount of information, Russell and Stephen, for instance, the nutritional content. There is a very important concern regarding, is this dredge, does the dredge contains contaminants? What about heavy metals? Are heavy metals preferentially bioaccumulated by the beans that are being grown that are growing in the dredge sediment? So all of those questions are being answered by Russell and Stephen. So what I have here in these figures are, I have the different treatments in the X scale, and I have in this specific one, the soil yield, and in the other one, the root biomass. So what I want to show you, and this is very important, because when we are adding an amendment, we want either a higher yield or we don't want any negative effects on the yield. And what Russell was able to prove is that when we are comparing the different treatments, the yield of the soybean did not decrease. When he did the statistics, there was no significant differences, but at least there was no a negative effect here. When we look at the root system biomass, the average of the biomass was increasing. So the more dredge, the more robust was the root system, which is a remarkable finding. Here in this slide, it's part of what Stephen is doing, and this is working in, it's still in the work. So Stephen is looking at the data and also doing a lot of statistics to try to understand if there is a preferential accumulation of heavy metals in the soybean grains. So here again, we have the different treatments, and here we have the heavy metal concentration or bioaccumulation in the beans, in the grains per se. And we have different elements, okay? If we look at, for instance, chromium, we can see that most of these error bars overlap, and potentially there is no preferential accumulation. So if we look at beans being grown with dredge sediment, there is an accumulation, but it's similar to that accumulation when we only have 100% farm soil. But more to learn from this from Stephen in the next couple of months. Okay, another experiment that is also in this mesocosm scale is an experiment being conducted by Siksha and Spencer. They are currently finishing their master's degree in my research. Both of them are, I am co-advising them with faculty in biology. And in this case, they were growing corn at also different ratios of their sediments. So we can see here their corn going in the greenhouse, and Stephen is actually adding a very nice addition to the experiment in which he is using millipedes as environmental indicators of maybe some sort of heavy metal bioaccumulation, let's say, for instance. So he's going to be studying if these millipedes are preferentially accumulating also heavy metals in their biomass. So some of the data that they have obtained so far, again, it's a very similar figure as the one that I already showed here. We have the different treatments, we have the corn yield, and we have the root biomass. We observe a very different, trends in this case, but as we are adding more dredge here, when you have 100% dredge, the yield was huge. Again, Siksha is doing statistics, so we don't have any statistic at the moment, but at least the addition of dredge sediment did not decrease the yield, with the exception of this example in this case. In overall, again, the root system biomass also increased as dredge sediment increased, and we have here a graphical representation of how that biomass system looked like. Okay, another experiment, it's being conducted by Oloshula here at BGSU, and Juan Pablo, he is from Ohio State University, also working with another collaborator there. But in this case, they are doing greenhouse experiment, but with specialty crops, and in a specific lettuce, strawberries, tomatoes, and carrots, because we need to expand the possibilities regarding how can we use, in what other crops can we use their sediments. So here are some data that they have shown so far. Here we have beautiful images of how the crops look like in the greenhouse and how their root system changed. So for this one in the upper portion, it's tomatoes. So we can see that, more robust root system when dredge sediment is added to the system. A similar trend was observed also for lettuce, and then for the carrots per se. In this case, that's a below biomass that we were observing. But both Juan Pablo and Oloshula, again, are working very hard on analyzing all the data, either agronomic data or soil health data. And Oloshula in specific, it's looking into the bioaccumulation of heavy metals, PCBs, PAHs, organic contaminants into the biomass of the different plants. So again, more to come from Oloshula in the near future. Now I want to jump to another scale. So now I am showing you a research at the farm field scale. And in this case, Siksha, once she graduates from her master's degree, she's going to be moving on and doing her PhD in biology with Dr. Jawishu and myself being her co-advisors. But in this case, now we have a farm field demonstration. So I want to see if some of the results that I have seen and observed in greenhouse experiment, do they translate to the field with way different environmental conditions? And now instead of a bucket, I have acres of land to see if this is possible or not. So this specific project is, the title for this is Beneficially Using Dredge Material as Farm Amendment to Improve Soil Health and Crop Yield, a demonstration farm project. Okay, so for this specific one, I rented land at the Agricultural Incubation Foundation here in North BGSU. And what I have here is the experimental design. We have 4 fields One, it's a conventional managed field, and I want to clarify that all of these fields are organic fields, so no pesticides and no synthetic fertilizers. The only amendment that was added was the dredge sediment at different rates. So we have the conventional with 0 addition of dredge sediments. We have 2 other fields that has 40 tons of dredge per acre and 80 tons of dredge per acre. And all of these received, they were planted with corn. An additional field received 64 tons per acre of dredge, but soybean was growing this specific one. The farmer also was able to look at crop yields on other farms, fields that did not contain dredge sediments too. So here it's a timeline that I want to show you of how things have been done for this specific experiment. So first, many different trucks were dumping dredge material into the area. I think that, I don't know, I will say that we have, I don't remember right now, maybe like 200 tons of dredge material was mobilized to the farm field. And then we used, for the first time in Ohio, at least to my knowledge, we used a manure spreader to spread the dredge, and it worked beautifully. So we was very happy that it was successful. Then the farmer tilled in the dredge. Then that was followed by making sure that he has all the preparation for planting. Then we went into the growing season and harvested, and all of this already has been harvested. All the soil samples and crop samples have been collected, and Siksha will be analyzing all of this data once she moves into her PhD. So here I have some data so far from this experiment. So here it's the Agricultural Innovation Foundation. Here are the different greenhouse units that I have rented in the past that my student has been working with. But here are the specific fields that I rented. So again, here we have that soy bean field where 64 tons per acre were added. And then I have 3 consecutive corn fields where 80, 0, and 40 tons of dredge sediments were added. And then additional soybean fields with no dredge added whatsoever. But what I want to show you in this specific slide is how the yield change when we place the dredge. So let's focus first on the soybean field. Okay, let's compare soybean field 1 with 2 and 3. And we can, what I want to compare is the yields. So for the field that had dredge, we had a 52 bushels per acre of soybean. And then for the fields that have no dredge whatsoever, we have 58 and 45. So it's kind of comparable. So we can conclude that, hey, the addition of dredge sediments, it's providing some nutrients, and we see no detrimental effects on the yield of soybean. However, we saw a different trend for the cornfield. So again, let's compare cornfield 1, 2, and 3 with 0 tons. In this case, the farmer measured 122 bushels per acre when 40 tons of dredge were added, 118. And when 80 tons of acre were added, 102. Again, there's no statistical data for this so far, but we can observe that the yield decreased. And this was a surprise and a frustration because we want to see no detrimental effect as we have been observing in the greenhouse experiment. So when I talked to the farmer, he explained to me that he believed this was this phenomenon was observed because when you are putting 80 tons or 40 tons of dredge per acre using the system that I just showed you, using this type of heavy equipment, the soil is being compacted. And then we also did it in May, which the soil was kind of squishy and wet. And the farmer believed that things might look different if we can add the dredge sediment in fall. So maybe the decrease in yield was due to the compaction of the soil because of the heavy machinery moving many times in the area to spread the dredge. But that can be corrected potentially if we spread in a different season, more towards the fall, that the soils are drained and are not so saturated with water, and they will be more susceptible to compaction. However, if we look at the phosphorus concentration in the fields before planting and after planting, and I have here all different fields, and I have the yields that I already discussed. But what I want you to see is the P value, the phosphate value, the bioavailable P before and after. So when 80 tons of 80 tons of dredge per acre were added, we can observe that before adding anything, the parts per million of the dredge was 21. After, it was 35. So we were able to amend and increase the phosphate by just adding the dredge sediment without adding any synthetic fertilizer. And the same was observed for the other treatment that we also put dredge sediments. So it is working regarding amending the soil with respect to the nutrients. We want to see if we can also get better yields if we place the dredge sediment in another time, within the year. So, and that is moving me to the next project that I have obtained. For this project, it's just out of the oven. It's just recently baked, and I'm going to have new master students that I'm going to be recruiting for fall 2023, in which they're going to be working with my next, version 2 of the experiment that I just showed you. So in this case, it's, again, I'm still in that field scale, but in this case, we are going to be adding to the equation hydrological budgets. And what does that mean? So now we are going to be collecting surface and subsurface water at each of the fields that are going to be amended with the dredge sediments. We're going to be looking also at the food safety, soil health, crop yields. But in addition, we want to know and understand, when we are adding the dredge sediments, are we enhancing the export of these nutrients into the waterways? Or will the dredge sediment provide nutrients, but they will be cycling in the soil in a way that plants and the microorganisms can get access and cycle them within the soil without losing them into the waterways. So that's what we're going to be doing in the next version of the other experiment. I'm working with Dr. Kevin McClooney in biology, Dr. Carlos Soto in SEAS, and also with some collaborators at Ohio State University. So again, we're going to be having conventional management and adding dredge sediments at different rates. And we are going to be looking at the whole set of data in addition to these hydrological budgets. Okay. So here are some examples of the edge of field that we're going to be putting in the fields. So we're going to have flumes to collect the surface water. We're going to have also weirs to measure the tile drainage flows, and we're going to be using ISCO samplers in both settings to collect water samples to measure the nutrient content that it's being exported into the waterways. So finally, the last scale that I want to showcase here is another project that also has been pretty much approved, or it's going to be approved potentially in March or April this year, 2023. And this one now it's putting in place different best management practices, but at a watershed scale. So in many instances, most of the research that has been conducted so far look at the field scale, but not doing a systematic implementation of BMPs at a larger watershed. So we are going to be doing a pilot watershed project with and Katarina, she's gonna be doing some of the experiments that I'm gonna be involved here, and I will tell you briefly what we're gonna be doing more. And I'm working with the Ohio State University, Dr. Jay Martin. He is the PI of the project. So this is a 16 million project that Dr. Martin is managing, but only a sliver, which is a really good sliver of piece of the cake, sliced piece of the cake, I'm gonna be getting to do my research. And I'm gonna be in charge of the soil health characterization across all the farms that we're gonna be studying in our experimental watershed, which is named Shallow Run. So the idea for this project is to implement in this watershed at least 70% of the land that is being used for farming to have some sort of BMP. Okay, and the idea is that based on modeling, if we implement BMPs at this percentage, there is gonna be a 40% decrease in phosphate export into the waterways. And this is the number that we are targeting to decrease or at least have under control the algae blooms that we have been observing in Lake Erie. Okay, so here are some examples of the BMPs that are going to be used in this specific project, again, at a watershed scale, but the one that we want to make sure that it's implemented to the higher degree is subsurface placement. Because it's known that if you broadcast the fertilizers, there's more chances for that fertilizer to be, if you have a water event, a rainfall event, to be dissolved and to be lost into the waterways, versus if you place it a few inches below the surface of the soil, and then those nutrients are going to be more readily bioavailable by the root system when we have crops in the area. So this is a very important project that many different collaborators are working with. It's a very complex project because we need to recruit many farmers in the area. We want to make sure that we have a communication, and we are showcasing and sharing all of the results with different stakeholders in the area. There's going to be a section of identifying first the farmers, characterizing the fields, having some sort of baseline, and then going into the implementation of all of the different best management practices. And then we also are going to be monitoring the soil health and the water quality. So this is a 5-year project that, it's a huge investment, and a lot of collaborators making sure that we can target a more holistic way of putting BMPs at a watershed scale to try to reach that reduction of 40% phosphate being exported into the waterways. So with that being said, that's the last portion of the research that I want to share with you guys. Again, you can see how I have been able to do research from so many different scales and aspects. And I want to say thank you to my master's students and the undergraduate students that have been working with me in this research, again, because I wouldn't be able to do any of this work without my students. And also here, I want to say thank you to all of these collaborators. So thank you so much for taking some of your time listening to this presentation. I hope that you enjoyed the presentation, and if you have any comments, you can email me back. So thank you so much. And I'm gonna just put the first slide here for just a second. So here is my email if you want to email me to learn more about my research. Thank you so much. Bye-bye.